Warm greetings to all. My name is Amrita Philip, and today I'm here to take a class on the microbial analysis of food. First of all, what is microbial analysis of food? It is a use of biological, biochemical, molecular, or chemical methods to detect, identify, or count the number of microorganisms present in a food item. Why is the microbial analysis of food such an important subject? Because food is a good culture medium, there is a possibility that a lot of microorganisms can grow and multiply inside the food product. The presence of microorganism in food causes the decomposition and spoilage of the food item. Therefore, microbial analysis of food is an important quality assurance of the food. And the microbial analysis of food gives an idea about the following concepts like the quality of the food, the sanitation condition of food, storage condition of food, types of microorganisms in food, and the quality of the raw materials used. There are various steps in the microbial analysis of food. The steps include the collection of the sample, the preparation of the sample collected, the transportation of the sample to the laboratory, and the microbial analysis of the sample itself. Collection of the sample. Collection of sample is an important step in the microbial analysis. The sample of food can be obtained from the processing line during the manufacturing, or from the storage house, or from the market that is directly purchased, and the sample could be representative and the sample should be the representative of the whole lot of the food. The sample should be collected randomly from a lot, and the outer surface of the container which is cleaned by washing or by using 70% ethanol. The sampling devices that is used include sterile spoons, pipettes, sterile scissors, knives, or can openers. Preparation of the samples collected. The preparation of the collected samples is different for both solid and liquid foods. For solid food, it is mixed with a sterile diluent in a mechanical blender to obtain a homogeneous suspension. For example, like if you are taking 10 grams of food, if you add 100 ml of diluent, then the dilution of this will be 10 raised to minus 1. And for liquid samples, we can serially dilute them to obtain the dilutions like 10 raised to minus 1, minus 2, minus 3, etc. Transportation of the collected samples. Transportation of the samples is also an important step because during the transportation, multiplication or the death of the microorganisms in the sample should be prevented, and the sample should be delivered to the lab as soon as it is collected. And the perishable or unfrozen samples, they should be stored in 0 to 5 degrees Celsius and transported in insulated containers so that there is no heat exchange. And the frozen products must be transported as it is in frozen condition, and it should be maintained in a frozen condition until it is taken out for analysis. Microbial analysis of food. Microbial analysis of food includes three major steps. The first one is the culturing method. It is the most traditional method used for the microbial analysis, and it is based on the culturing, enumeration, and isolation of presumptive colonies for further identification. And if necessary, the food samples that is collected is homogeneous, concentrated, or pre-enriched prior to the culturing. After all these methods, these pre-treated samples, they are plated on either a non-selective media, on a selective media, or on a differential media. And there are various types of culturing techniques available, some of which are total viable count, pot plate method, spread plate method, agar droplet method, etc. Second type of microbial analysis is the immunoassays. It is based on the specific binding between the antigen and antibody. Usually, these techniques are used for the identification of diseases in clinics or for clinical diagnosis of diseases. But these are becoming increasingly popular for the rapid detection of the microbes in food. And the types of immunoassays include enzyme-linked immunosorbent assay, ELISA, immunoprecipitation assay, immunodiffusion assay, immunofluorescence microscopy, etc. The third method is the polymerase chain reaction or the PCR method. It is a method in which the amplification of nucleic acid target sequence takes place. The targeted sequence that is amplified may be either specific genes or a repetitive areas in the nucleic acid. Uh, Usually, the common target areas are like the genes that code for virulence factors or that code for toxins or cellular metabolites. The PCR techniques are also developed for identifying genetically modified organisms and their derived materials in food. Often the PCR after the often after PCR visualization is using gel electrophoresis or using specific nucleic acid probes. These probes they may be radioactively labeled, and while binding and hybridization we can visualize it using autoradiography. With this, we have come to the end of this session. Thank you all.